What is malaria? Malaria is an infectious disease transmitted by Anopheles mosquitoes. The mosquitoes themselves are not parasitic, but rather, some of them carry Plasmodium parasites. Once bitten, these parasites enter the human bloodstream and attach themselves to the liver. After attaching to the liver, the parasites multiply before re-entering the bloodstream. Due to the stage of multiplication, symptoms of malaria could take anywhere from 1 to 4 weeks to appear. The symptoms can range, but typically resemble the flu with headaches, vomiting, and fevers. While there is a cure, when malaria isn't treated soon enough, it can result in anemia and even death. In many rural African areas, people infected with malaria often don't have the time to get necessary medical treatment due to the severity of the disease.
Earlier studies have shown that the bacteria contained within the gut microbiome help control how cells respond to viral infections like malaria. The study conducted by Villarino and others shows the correlation between gut microbiota and severity of malaria in mice. Genetically similar mice populations collected from different vendors were separated into 4 groups. Villarino and colleagues then infected each of these groups with multiple species of Plasmodium parasites, P. yoli, P. chabadi, and P. burgei. Once infected, researchers tracked their progression and noticed that half of the mice developed severe malaria and even experienced death, while the other half developed less severe malaria without any death. The resistant mice were shown to have higher concentrations of Bifidobacteriacea, Clostridiacea, Rhizopelotracea, Lactobacillicea, and Peptotreptococcacea. The scope of the study was then narrowed down to 2 mice populations, JAX and NCI. These 2 populations of mice showed the greatest disparity in microbial communities throughout small intestine, cecum, and plasma. In order for Villarino and his fellow researchers to assure that the variants of microbial community could be associated with the development of malaria, they transplanted part of the intestinal system of germ-free, genetically identical mice with the intestinal system of JAX or NCI. After confirming that the transplant altered the microbiome of the germ-free mice to match that of JAX and NCI mice, Villarino and fellow researchers infected the germ-free mice with a P. yoli parasite. Following infection, the mice transplanted with JAX cecums had similar symptoms to the JAX mice, and those transplanted with NCI had similar symptoms to the NCI mice. This discovery confirmed the researchers' hypothesis that microbiota affected malaria severity. Villarino and colleagues also hypothesized that diet had an effect on the gut microbial community and thereby the resistance to the severity of malaria. Of the bacteria found in resistant mice, researchers chose to isolate Lactobacillus and Bifidobacterium. JAX and NCI mice were separated into groups and treated with antibiotics, yogurt that contained Lactobacillus and Bifidobacterium, or a mixture of antibiotics and the yogurt. Researchers saw a decrease in the effect of malaria in susceptible mice when treated with antibiotics and yogurt, supporting that Lactobacillus and Bifidobacterium could directly affect malaria severity.
The results of this experiment reflect the need for further ecological studies of malaria and the associative gut microbiome. Within the large field of biology, these experiments shed light on the impact of basic concepts such as species niche, adaptation, and environmental effects. Each species of the gut bacteria had their own fundamental niche. However, when Lactobacillus and Bifidobacterium were present or added to the microbiome, up to 4 species of bacteria nearly disappeared, while other previously scarce species thrived. The presence of Lactobacillus and Bifidobacterium have shifted the fundamental niche of the other bacterial species to a much smaller realized niche. These species shifts were crucial in being able to alter the gut microbiome to one that is more resistant to severe malaria. Africa happens to be the perfect niche for Anopheles mosquitoes, providing them with a non-resistant food source as well as a proper climate. Secondly, the African Anopheles did not make the same adaptations as the American Anopheles, and are still carriers of the Plasmodium parasite. The American Anopheles were able to develop resistance to the parasite through allopatric speciation, which is where a species is split into 2 populations that have a geographical barrier and ultimately form 2 separate species. The children in rural African communities have been found to have extremely low levels of Lactobacillus and Bifidobacterium when compared to European children. Such a difference is a result of malnutrition, which is an environmental factor. While the odds seem to be against Africa, there is still hope. The importance of this experiment is to demonstrate that a change in diet alone can impact the gut microbiome, altering it for the better.